Sairam students, in this class we will study about the unit 14, that is biomolecules. So biomolecules means what? What and all we will study in the biomolecules. It's very amazing thing that the living system which is made up of non-living system, means non-living molecules. What are those non-living molecules? For example, you can take a carbohydrates which is present in our body, that is a non-living thing. But we are living by, there are so many molecules which are present in our body which are very essential to live. For example, see what in all chemically happen in our body, it will come under biochemistry, means the bio is one side and chemistry is one side. So together we will study, that is biochemistry. So for example, you can see carbohydrates, proteins, vitamins and nucleic acids, these all are present in our body, but those are chemicals, means those are non-living materials, means molecules. Here proteins and carbohydrates are very essential for our living things. So now we will, this class, it means in the whole chapter we will study about the proteins, carbohydrates and nucleic acids, vitamins and so on. So the objectives, what are the objectives of this chapter? So we will study about the introduction of biomolecules, carbohydrates, proteins, enzymes, vitamins and nucleic acids. We will study about here the whole, what are the functions and what are the preparations and how it will act in our body, we will study in detail. So next one, that is introduction. So living system grows and sustains and reproduces itself. The most amazing thing about a living system is that it is composed of non-living and molecules. The pursuit of knowledge of what goes on chemically within a living system falls in the domain of biochemistry. Living systems are made up of various complex molecules like carbohydrates, proteins, nucleic acids, lipids, etc. Proteins and carbohydrates are essential constituents of our food. So next one, see here, biomolecules means again we divide it into two parts, that is the macromolecules and micromolecules. What is meant by micromolecules? The biomolecules whose molecular weight less than 1000, for example sugar, lipids and vitamins. And what is meant by macromolecules? Macromolecules means little bit bigger molecule. The biomolecules whose molecular weight greater than 1000, for example protein, polysaccharides and nucleic acids. And here again we will see biomolecules, we divide it into two types. Here micromolecules, micro means very small molecule, macro means bigger molecule. And here this molecular mass is less than 1000, molecular mass is greater than 1000. Okay. So for example, you can give here a carbohydrate, sugar, okay, and all. And here you can give the example polysaccharide. Polysaccharide means the bigger than this molecule. Now we will start with carbohydrates. What is mean by carbohydrates? Carbohydrates means it is called hydrates of carbon. Carbohydrates are primarily produced by plants and form a very large group of naturally occurring organic compounds. So general formula CxH2OY. For example, glucose, that glucose formula is C6H2O6, means we will write as C6H12O6. Acetic acid, as we know acetic acid formula is CH3COOH, fits into general formula but it is not a carbohydrate. Chemically, the carbohydrates may be defined as optically active polyhydroxyaldehydes or ketones, or the compounds which produce such units on hydrolysis. Carbohydrates, they are sweet in taste and they are also called as a saccharides. The word came from Greek, that is saccharon means sugar. So here see carbohydrates. So we will first study about the carbohydrate. Carbohydrates means the hydrates of carbon. Hydrates of carbon. So, the general formula is C6H2OY. Okay. So, here X and Y, there is a components here. So, we can write the example here C6H12O6. This one we will write as a C6H2O. Sorry, this is 6. Okay. See, here X is 6 and here Y is 6. Now, for example, if you take the acetic acid, general formula is CH3COOH. Okay. And here it will just fit into this general formula. The carbohydrate general formula is C6H2OY. And even CH3COH is also fits into this formula, but CH3COH is not a carbohydrate. It is not a carbohydrate here. It is very important to know, and there are few compounds which fits into general formula, but they are not carbohydrates, and carbohydrates, all carbohydrates will fit into this general formula. This is CXH2O and Y, and even carbohydrates is also called as saccharides. Saccharides, the word came from Greek, that saccharon means that is a sugar, and always the carbohydrates are sweet in taste. Okay. For example, you can take a sugar is always a sweet in taste, and even sucrose and fructose, all are sweet in taste. So, now we will study about the classification of carbohydrates. So, classification mainly we divided into 3 types based on the hydrolysis. The first one is monosaccharides and oligosaccharides and polysaccharides. And again monosaccharides, again we divide into 2 types, I will explain later, that is aldose and ketose. And oligosaccharides, again we divided into, that is 2 to 10 means 2 to 10 means there is 8 types are there. Carbohydrates that yield 2 to 10 monosaccharide units on hydrolysis are called oligosaccharides. Polysaccharides means more than 10 units will be there. So, now we will study in detail the classification of carbohydrates. So, carbohydrates mainly we divided into 3 types based on the hydrolysis, based on behavior of hydrolysis. Hydrolysis means when we put the compound into water, it will give the product, next the product, right. So, we have to classify the carbohydrates on based on the hydrolysis. First one is monosaccharides, and next one is oligosaccharides, and last one is polysaccharides. So, first one, what is mean by monosaccharides? Monosaccharides means when you put into the water, when you hydrolyze, there is no further reaction takes place. It means only one kind of monomers, means monosaccharide is present inside the carbohydrate. For example, you can take a glucose. Glucose, what is the molecular formula? C6H12O6. There is only one monosaccharide. If you put into the water, it will be remains as same it is. So, it will not further hydrolyze. This is even you can take the C6H12O6. It is not a glucose, it is a fructose. Okay. Why? Though both having same molecular formula, but they have a different structure. That is why they both will be having same molecular formula C6H12O6, and if you put this into the water, they will not further hydrolyze. And next one is oligosaccharide. Oligosaccharide means when you hydrolyze oligosaccharides, it will give 2 to 10 monosaccharides. Okay. Those again we can call as a trisaccharide, tetrasaccharides. First one is disaccharide. Disaccharide means 2 to 10, right? Disaccharide, trisaccharide, tetrasaccharide, etc. Okay. Di means 2. Okay. Tri means 3. Tetra means 4. Penta. It's like till 10. 2 to 10 it will give the monosaccharides. When you hydrolyze the oligosaccharide compound, it will give 2 to 10 monosaccharide compounds. For example, in oligosaccharides, most of we will study about only disaccharides. Okay, disaccharides we can give the example, that is sucrose. So when you hydrolyze this one, sucrose, we will get glucose plus fructose. Okay, it means when you hydrolyze the sucrose is a disaccharide, it is having a 2 compounds, that is glucose and fructose. Okay, when you hydrolyze we will get the two compounds. Like that, trisaccharides and tetrasaccharides, there are so many are there, but only we will study most of the compounds in the disaccharides, in the disaccharide form. So next one is the polysaccharides, more than 10 monosaccharides present in this. When you hydrolyze, more than 10 monosaccharides is present in that compound. So that is called polysaccharides. Okay. So next again we will study, again carbohydrates is divided into 2 types. Okay, that is reducing sugar and non-reducing sugar. So what is mean by reducing sugar? The carbohydrates which are reduced Fehling solution and tolerance reagent. So for example, all monosaccharides, both aldose and ketose here. So non-reducing sugar means the carbohydrates that reduce neither Fehling solution nor tolerance reagent. For example, all disaccharides except sucrose, because aldehydic or ketonic groups are bonded in sucrose. Here it is very important even for the examination. So carbohydrates again we divide it into 2. One is reducing sugar, and one more is non-reducing sugar. So reducing sugar means it will reduce all Fehling solution and tolerance reagent. So these are 2 tests. Okay, usually these tolerance solution or Fehling solution and tolerance reagent, there are 2 tests. When you react the carbohydrates with these 2 compounds, there are solutions will be there, you just react with that and it will give the reaction there, and we will get the silver color PPT in the test tube. And you just add a carbohydrate, take a carbohydrate in a test tube and carbohydrate with just add a Fehling solution and tolerance reagent, it will give the reaction. That means the carbohydrate just reduce on Fehling solution and tolerance reagent. Whatever the carbohydrates, if it reduce on Fehling solution and tolerance reagent, those are reducing sugars. And what is meant by non-reducing sugar? And here the non-reducing sugars are, it will not reduce either Fehling solution and nor tolerance reagent. It means it will not give any kind of reaction there. Suppose if you take carbohydrates in the test tube, just take a test tube, okay, just a carbohydrate here and just add a Fehling solution and tolerance reagent here. It will not give any kind of reaction there. If it is not giving any kind of reaction means those are non-reducing sugar. If it is giving the reaction means, if it is giving any kind of reaction means that those are the reducing sugars. Very important for the examination. And here the all kinds of all the kinds of monosaccharides here, and here for example disaccharides except sucrose. This is very important to know why only sucrose will not give the reaction. Here in sucrose both aldehydic and ketonic groups are bonded with each other. For example, the sucrose I told you, right, it is a combination of glucose plus fructose. Okay, glucose is having an aldehydic group and fructose is having a ketonic group. There the aldehydic and ketonic groups are bonded with each other. That's why it will not give the reaction on Fehling solution and tolerance reagent. And next, see again carbohydrates, again we classified as aldose and ketose. So what is mean by aldose and what is mean by ketose? Here again we divided aldose and ketose. We know, all of you very familiar with the functional group, that is the aldehyde and ketone. So, what is the functional group of aldehyde? That is a RCHO. Usually we will write general formula RCHO, and here RCO R dash. This is CO is the ketonic group, here CHO is the aldehydic group. See the monosaccharide which is having aldehydic functional group, that is called aldose. Suppose if monosaccharide is having ketonic group as a functional group, then that is called as a ketose. Okay, it will come under carbohydrates, usually in the monosaccharide. Okay, monosaccharide. So in monosaccharide means aldose and ketose. Aldose means the monosaccharide which is having a aldehydic group, that is a CHO, and monosaccharide which is having a ketone as a functional group, that is a ketose. Okay. Now different types of monosaccharides we will see. So see carbon atom general term and aldehyde and ketone. See if carbon atoms are 3 in the compound or in the molecule, the general term is triose. Tri means 3. If it is having aldehydic group, that is a aldotriose, and if it is having a ketone group, that is a ketotriose. And next is if monosaccharide is having carbon atoms 4, then general term is tetrose. Tetra means 4. So if the monosaccharide is having aldehydic group, that is a aldotetrose, and if it is having a ketonic group, that is a ketotetrose. And if 5 carbon atoms are present in monosaccharide, that is a pentose, that is general term, and if it is having a aldehydic group, that is a aldopentose, and if it is having a ketonic group, that is a ketopentose. Next one is a carbon atom 6, that is a hexose, general term is hexose. Then if it is having aldehydic group, that is a aldohexose, and ketonic group means ketohexose. Next is carbon atom, if it is having carbon atom 7, then it is a heptose. Then aldehydic group, that is a aldoheptose, and ketonic group means that is a ketoheptose. So this is all about the different types of monosaccharide. Okay, 3 to 7. Next we will study about the preparation of monosaccharide. Here in monosaccharide usually we will study glucose. Glucose is very important for even your life, right? So glucose occurs freely in nature as well as in the combined form. It is present in sweet fruits and honey. Ripe grapes also contain glucose in large amounts. So first preparation of glucose, first one is from sugar, or else it is a sucrose. Sucrose is present in cane sugar. If sucrose is boiled with dilute HCl or H2SO4 in alcoholic solution, glucose and fructose are obtained in equal amounts. So reaction you can see there. Okay, see here, sucrose is having general formula C12H22O11. Then when we boiled with that, why always we will write H plus. What is mean by H plus? If we are boiling with HCl or H2SO4, we know those will easily give the protons H plus. H plus means always that is the acid, right? So when we boil with acid, that is HCl or H2SO4, anyone you can take. So when we boil sucrose, then we will get the glucose and fructose. You can see the structure here. So both are having same molecular formula, right? But they are having a different structures. We will study in later. So when see here, it is a balance. Here see there is C12 are there, C6 and C6 there, H only 22. So here we are reacting, we are boiling with water, right? So the H here too there are total, if you count the H12 here, H2 there are total 24, right? So here only 11 oxygen is there, 1 oxygen again it will take, then OX and O6 and O6. So when you just boil sucrose, okay, it will give the glucose and fructose. Okay, this is first preparation from the sucrose which is present in cane sugar. So next preparation is from starch. Okay. Commercially, glucose is obtained by hydrolysis of starch by boiling it with dilute H2SO4 at 393 Kelvin under pressure, 2 to 3 atmospheric pressure. So, starch or cellulose, okay, the molecular formula C6H10O5, N number, because starch is a polysaccharide, right. So, NH2O, N number of H2O, then it gives the N number of C6H12O6. Okay. So, you can see this is the starch or cellulose. When you react with water, when you boil, we will get the glucose. Means this reaction only we will prepare commercially. Commercial means industrial level. In large quantity we can prepare the glucose by taking starch. Okay. So, this is all about monosaccharide and preparation of the glucose. So, in next class we will discuss about the structure of glucose. Thank you.